Boxwood is a centerpiece and common component of gardens and historical sites throughout the Mid-Atlantic region of the United States. The plant remains a very popular choice in landscapes today, with newer varieties known for their resistance to disease and pest issues, and the general tolerance of boxwoods to drought. Deer also don't seem to browse on them often. In September 2011, a new disease of boxwood called Boxwood Blight was reported for the first time in North America. Currently, the Boxwood Blight pathogen has been reported with isolated finds in 10 states and 2 Canadian provinces. Prior to its find in North America, the disease was known to be distributed throughout Europe and New Zealand. Boxwood blight can be traced back as far as the early 1990s in Europe. In 1994, a cylindrocladium species was isolated from defoliated boxwood plants by scientists at the Royal Horticultural Society. Then, again in 1998, a cylindrocladium outbreak was discovered in the UK and New Zealand. This time, researchers in the UK and New Zealand determined the cylindrocladium on the boxwood was a newly described species. In 2002, the newly described species was named and described by Pedro Kraus as Cylindrocladium pseudonaviculatum. This name is based on fungus vesicle morphology. Patrice Hendricott also named the fungus in the literature as Cylindrocladium buxicola. This is a name based on host. Both names are found in the literature currently, but Cylindrocladium pseudonaviculatum is technically the proper name as it was published in the literature first. Cylindrocladium is a genus of fungi comprised of 52 recognized species. They are related and share morphological characteristics of cylindrical spores, vesicles, and they often form resting propagules called chlamydospores and Microsclerotia in host tissue. The cylindrical spores produced by this group of fungi are adapted for short distance dispersal between plants. They are spread by wind-driven rain or water splash from overhead irrigation, but they may also stick to animals, tools, and equipment. Vesicles are another microscopic structure produced by this genus. Vesicle morphology is useful for species identification in the genus Cylindrocladium. Microsclerotia are another common structure produced by this genus. These structures are formed in host tissue and allow the fungus to survive in soil for as many as 15 years. Cylindrocladium species are important plant pathogens. They impact ornamentals, field crops, and forest seedlings. Azaleas, spruce, peanuts, and eucalyptus are just a few examples of crops that are often afflicted by this pathogen. Symptoms of Cylindrocladium include root rot, cutting rot, leaf spots, cankers, and shoot blight. Although most Cylindrocladium pathogens do cause a root rot, the Boxwood Blight pathogen appears to not infect the roots of the boxwood. From what has been documented in the field, the Boxwood Blight pathogen infects all boxwood species and cultivars important to the horticultural industry. This list here represents cultivars infected naturally in landscape and nursery settings. Here is a continuation of the list of natural hosts. The take-home message is currently all cultivars of boxwood appear to be quite susceptible to the Boxwood Blight pathogen and develop significant symptoms when infected. Interestingly enough, most of the diversity within the genus Buxus is in Southeast Asia and the Caribbean. No one knows where the Boxwood Blight pathogen originated from, but often pathogens that have the ability to infect a specific group of plants sometimes originate from the center of origin of that plant group. A great example is late blight of potato. This pathogen seems to have originated from the Andes Mountains, which is the center of origin of the potato. Although within the boxwood family, the greatest diversity is in the genus Boxus. The boxwood family also includes Pachysandra, Sarcococa, and Styloceras. This has prompted the question, are other genera within the boxwood family hosts? So far, 2 other genera have been infected by scientists in the laboratory. Here is a picture of Pachysandra infected by researchers from Connecticut. So far, Pachysandra has not been found to be a host outside of the laboratory, and its relative susceptibility as compared to boxwood is not well understood. Still, Pachysandra should be considered a potential host of the Boxwood Blight pathogen. Sarcococa has also been infected by researchers under laboratory conditions in the UK and Virginia. It has been infected, but appears to be less susceptible than the boxwood. Boxwood blight can develop fast. Based on interviews with growers and homeowners, within a few weeks a crop or a landscape can go from looking healthy to being severely impacted. It is important for nurseries to scout and find boxwood blight at its early stages so as not to ship, sell, or move symptomatic plants to help slow the spread of this new disease and protect clients and customers. Here, plants have an advanced stage of the disease. These plants are clearly defoliated. The leaves have fallen off. This is characteristic of boxwood blight, as defoliation does not always initially occur with the onset of other diseases. Often with other ailments, straw-colored leaves will remain attached to the plants for months. More defoliation shown here on rooted cuttings, afflicted with boxwood blight. And here are some plants with almost 100% defoliation. This occurred within one growing season between June and October of 2011. Another symptom often associated with boxwood blight are narrow blackish brown lesions. This here is a branch with these small cankers. These symptoms are a good early indicator and may sometimes be present before advanced defoliation. On occasion, other fungi may cause similar symptoms. Remember to always call your Department of Agriculture or County Extension to get samples tested in a lab. Here are more advanced elongated cankers shown on American boxwood. Leaf spots at the early and more progressed stages of this disease are common. The leaf spots often have a dark zone around the edge of the leaf spot as shown to the left. This dark zone is not always apparent on smaller spots caused by this disease, as shown to the right. After leaves are infected, plants will defoliate within a span of weeks under the right environmental conditions. On the right, the leaves are about to fall off. Note they are not retaining straw color, as is typical with other ailments of boxwood. Here are defoliated plants in a landscape with severe boxwood blight. The plant circled in red was planted in 2011 and likely introduced the disease. Conditions were wet and mild and favored disease development. Spore masses are produced during warm, moist conditions. They are stellate, referring to how the branches that hold the spores radiate out like a star. With some imagination, one can imagine these clumps of spores as individual stars. For the trained eye, this type of sporulation is distinct from other fungi that are found associated with boxwood. Like its relatives, the boxwood blight pathogen produces cylindrical spores adapted for relatively short distance dispersal between plants. Once the pathogen is introduced to a site, these propagules allow for infection of susceptible host tissue. Again, these spores are spread by wind-driven rain, overhead irrigation water splash, or infested equipment. One adaptation enabling soil-borne plant pathogens to survive in soil is the formation of specialized resting spores called Microsclerotia. Species of Verticillium, Rhizoctonia, and cylindrocladium all produce these specialized structures. Cylindrocladium species have been reported to survive as long as 15 years in soil as Microsclerotia, creating a challenge for managing this group of pathogens. In relation to the boxwood blight pathogen, One study conducted in England demonstrated the ability of cylindrocladium pseudonaviculatum to survive in infested leaf material as long as 5 years. However, authors did not observe any Microsclerotia and suggested the pathogen survives as mycelia. Based on these reports, if the Boxwood Blight pathogen had indeed lost or never evolved the ability to produce Microsclerotia in host tissues, one could infer that the pathogen may be less equipped for long-term survival in soil than other cylindrocladium species. Based on these assumptions, a study was conducted to determine the potential role, if any, of Microsclerotia in the life cycle of the Boxwood Blight pathogen. Unfortunately, we are finding that cylindrocladium pseudonaviculatum produces abundant Microsclerotia in host tissue. These specialized structures are ecologically important for this species and may contribute to its ability to survive in soil for extended periods of time. Soil flaming has been conducted in an attempt to reduce inoculum loads of infested soils at fields in Virginia. At one of these field sites, soil was sampled before and after soil surface flaming. Soil was separated into particle fractions and plated on selective media to evaluate inoculum levels before and after flaming. In the larger fraction sizes, it was clear that soil surface flaming, when carried out before leaves become incorporated into the soil, greatly reduced infested leaf debris. The top row here shows soil before flaming. The bottom row shows soil after flaming. Fragments of infested leaf were found in the flamed and non-flamed soil. However, there was significantly more colony-forming units in the soil before it was surface flamed, suggesting soil surface flaming will reduce inoculum levels in soil if conducted thoroughly. It is important to note that most of the colony-forming units found were present in the larger fraction sizes, which consist of leaf debris. It is thus important to flame the soil surface before leaf debris become incorporated into soil by erosion or before leaves are blown to adjacent fields by wind. Now, we're only talking about fields where the Boxwood Blight pathogen has been found. It is not recommended that one invest in surface flaming soil if the Boxwood Blight pathogen has not been detected at a site. So to briefly summarize the biology of this disease, in general, the Boxwood Blight develops and spreads during humid, warm conditions. The natural host range is limited to the Boxwood family. Spread between plants and within fields occurs by spores, which splash by wind-driven rain or overhead irrigation. Spores may also stick to clothes, tools, and animals, and other equipment. Long-distance dispersal occurs by moving infected plant material, contaminated soil, or muddied tools and equipment and tires. High populations of parasitic nematodes can stunt and kill boxwood. Note the picture in the upper left. The foliage has not fallen off but remains straw-colored after the plant dies. This is in contrast to boxwood blight, which typically causes defoliation. On the left is a common foliar leaf spot and dieback of boxwood called macrophoma. Macrophoma does not cause defoliation as rapidly as boxwood blight, and the fruiting bodies can be seen as dark spots as compared to the stellate sporodocia of boxwood blight. Macrophoma does not typically cause the discrete circular leaf spots either. Volutella is the most common fungus associated with blighted foliage of boxwood. It may act as a pathogen, but is frequently a secondary saprophyte on boxwood as well. During moist weather, the fungus forms clusters of salmon pink spores on the underside of leaves. The spores are seen as dry crust on the underside of the boxwood leaves long after spores dry out. Typically, Volutella does not cause defoliation. Dead straw-colored leaves will still remain on the plants for the season. Both Volutella and Macrophoma are common and frequently found on boxwood infected with boxwood blight. The presence of one or both of these pathogens does not rule out the presence of boxwood blight. For the rest of this presentation, we will look at best management practices aimed at reducing the potential of boxwood blight to impact your business. The topics covered will include exclusion, water management, sanitation, inspection, training, record keeping, and traceability. Exclusion for producers can best be achieved by propagating on-site from cuttings harvested on-site, not bringing in any boxwood material. This would include using mother plants that have been in your possession for several years or were grown on site. Limit access to mother plants and isolate them from traffic and other stock. Monitor mother plants regularly and don't use fungicides on mother plants. If you are a retail operation or you decide that you need to bring in boxwood material, buy from suppliers who have entered a Boxwood Blight cleanliness agreement. Create an isolation area for new materials and returns. This area should be separate from other host material by 3 meters or a physical barrier. It should be swept up and kept free of host debris weekly. Also, new host material should be held here for 30 days so they can be inspected regularly before being released into the general nursery area. Also, this isolation area should be where all new shipments are unloaded of host material, and where all incoming hosts are inspected as or before they leave a truck. Any symptomatic material should be rejected and reported to the supplier. Also, it's important not to apply any fungicides in this isolation area. So this is a schematic of the flow of host plant material utilizing the isolation strategy. So to the far left, anything in orange represents new host material. It's held in an isolated area away from the general population of the plants in the nursery. Plants are held here for 30-day observation, during which no fungicides are applied to them. They're under enhanced sanitation of host material, and there is 3 meters between all blocks of plants. Plants should probably be grouped by each shipment. You could group them in other ways as well, but by shipment is one recommendation. This is where all the plants are unloaded and where all the potentially dirty plant material is held under isolation before it's declared clean and entered into the general population of the nursery. And anything in green represents accepted or material that has been approved to be released into the general population. Now, to the far right, it's important to consider the potential of landscape vehicles, or if it's a retail operation, even the general public bringing in plant materials that have been in the landscape or tracking in potential disease into your nursery. So you should have a loading area where all the plant material can be loaded onto trucks or into cars rather than allowing customers to drive where the host plant material is being grown and propagated inside your nursery operation. Know the signs and symptoms of boxwood blight and scout for it continuously throughout the growing season. Also, keep detailed records of plant material, where it's come from and where it is going. Avoid overhead irrigation of host plants when practical. When using overhead irrigation, irrigate so as to reduce leaf wetness, period. Also, monitor for host plant debris and water runoff. Minimize water runoff such that it does not run from one host plant production area into another. As much as possible, avoid standing water in and around plant blocks. Cleaning leaf and soil debris from host blocks is very important. It helps if you have the plants on a surface that is easily cleaned, such as asphalt or weed mat. Here's an example of a container on bare ground. This is quite hard or impossible to clean, and the leaf debris will become incorporated into the bare soil rapidly. Weed mat over gravel allows for good drainage and easy cleaning with a broom, much better than having plants stored on bare ground. Clean equipment, tools, and boots with 70% ethanol, 1 to 10 bleach, or contrary ammonia after working with host material. Keep in mind that you must first rinse off soil and debris before you clean the equipment with any of these sanitizers in order for them to work effectively. Lastly, if you do get boxwood blight, destroy plants within the infected block. It also will be helpful if you have the plants grouped into blocks separated by 3 meters to reduce plant losses due to destruction associated with regulatory action. Also, act quickly and eradicate inoculum embedded in host plant debris. This can be accomplished by raking up and destroying plant debris, or potentially using an agricultural flamer to reduce the inoculum levels.